Welcome. Today, we're diving into the phlebotomy order of draw, a crucial step for anyone drawing blood. Whether you're a student or a healthcare worker, understanding the right sequence helps prevent errors and ensures accurate test results. Let's break it down together, step by step. By the end of this video, we're going to cover some key points. First, we'll define what the order of draw actually means. Then, we'll explore why it's so important in practice. We'll also learn to identify the common tube colors you'll encounter. And finally, we'll make sure you can recall the correct sequence confidently. These learning objectives will guide us through everything you need to know. Let's talk about the order of draw, the sequence in which blood collection tubes are filled. Following this order is crucial because it prevents contamination between additives in the tubes. That, in turn, ensures the test results are accurate and reliable. So, understanding the order of draw isn't just a technical detail. It's a key step in delivering trustworthy lab results every time. Let's talk about why this matters. First, it prevents additive carryover. That's when residues from one sample contaminate the next. It also improves specimen quality, making sure each test is as accurate as possible. Most importantly, it protects patient safety. When we get these steps right, we reduce errors and keep everyone safer. That's why this process is so critical. Let's talk about blood culture bottles, a key tool for detecting microorganisms in the blood. Each bottle contains an additive called SBS, which helps preserve the sample. We use two types, one aerobic, which needs oxygen, and one anaerobic, which doesn't. Both bottles must be collected under sterile conditions to avoid contamination. This slide highlights tube number 1, specifically for blood cultures, which plays a crucial role in diagnosing infections quickly and accurately. Let's talk about the light blue tube. It contains sodium citrate, an additive that's key for certain blood tests. It works by binding calcium to prevent clotting, so the sample stays usable. Common tests here include PT, INR, and PTT, all important for assessing how well blood is clotting. You'll usually see this as tube number 2 in the sequence. It's a small detail, but it makes a big difference in accurate results. When it comes to blood collection tubes, red and gold are the go-to choices for several key tests. We use them primarily for chemistry tests, hormone tests, and lipid panels. These tubes are often labeled as tube number 3, and you might see them called SSD tubes. They're designed to help separate the serum from the blood, making analysis smoother and more accurate. So, red and gold tubes play a crucial role in getting reliable lab results for these important health markers. The green tube contains heparin as an additive. We use it mainly for chemistry testing, which covers a wide range of blood analyses. It's also the go-to for some stat tests, those urgent ones that need quick results. In the order of draw, it's tube number 5. Understanding where and why we use this tube helps keep our testing accurate and efficient. The lavender tube is a staple in many labs, thanks to its additive, EDTA. This additive helps preserve the blood cells for accurate analysis. It's commonly used for tests like the complete blood count, or CBC, HbA1c for diabetes monitoring, and blood film examinations. You'll find it as tube number 6 in the sequence, ready to provide crucial insights into a patient's health. This slide introduces the gray tube, which contains additives like sodium fluoride and potassium oxalate. These help preserve the sample for specific tests. The most common tests we run with this tube are glucose and lactate. You'll often see it labeled as tube number 7 in the sequence. It's an essential part of the process when accurate measurement of blood sugar or lactate levels is needed. Here's a simple mnemonic to help us remember the key elements. BRAVE stands for blood cultures. Light blue reminds us of loins. Red and gold represent run. Green means great. Lavender is for long. And gray stands for games. It's a quick way to keep these colors and terms clear in our minds as we review the material. Mnemonics like this make complex information easier to recall when we need it most. Let's take a moment to check our understanding with a quick practice question. Which tube do you think is commonly used for a complete blood count test? Is it the green, the gray, or the lavender tube? The correct answer is the lavender tube. This is a key detail to remember when preparing samples for CBC testing, so keep it in mind as we move forward. Let's talk about some common mistakes we often see. Drawing things in the wrong order can throw off the whole process. Then, underfilled light blue tubes can cause issues with accuracy. Poor mixing of tubes leads to inconsistent results. And finally, incorrect labeling, that's a simple error with big consequences. Catching these mistakes early helps keep everything running smoothly. To wrap up, there are 4 key practices we need to keep front and center. First, always follow the correct order of draw. It's essential for reliable samples. Next, prevent contamination at every step, no shortcuts here. These steps directly improve patient safety, which is our top priority. And finally, they ensure the laboratory results we rely on are accurate and trustworthy. Together, these practices form the foundation of quality care. Thanks so much for watching. If you found this useful, please like, share, and subscribe to stay connected. Your support means a lot and helps us keep creating content that matters. Looking forward to seeing you again soon. Take care. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.